Hello everyone, my name is Aleksander Jankowski and I am presenting a poster called Go Functional Annotation of Genes in Chromatin Contact Pairs. What's our idea? You might be familiar with the standard gene ontology enrichment analysis. For example, in a situation when you have a set of genes of interest and you would like to tell whether these genes have something in common, more likely than by random chance, so whether this set of genes is somehow enriched compared to, let's say, the universe of all genes in a particular species. But sometimes it could be that the set of genes that you are having is coming with some structure. For example, you might be working with the data on chromatin conformation. I have an extract of a high-C contact map over here. It gives a fragment of a chromosome along X and Y axis, and the color intensity indicates the contact frequency. We see some regions that tend to have more often interactions inside than to the outside, called topologically associating domains. And you also see some peaks in these contact maps, which correspond to so-called chromatin loops. So regions in the genome, which could be quite far away in terms of linear distance along the chromosome, but they are brought together in close proximity and often have some interacting regions, like interacting enhancers and genes, target genes, or interacting pairs of genes. So when we have such set of pairs of genes, you might be asking a different question about enrichment of gene ontology terms. You might be asking whether the genes that are at both ends of these pairs have something in common more often than by random chance. And the central panel over here gives some details how our method works. Let me discuss it in detail. Let's assume that we have a set of gene interactions. It's represented as a graph. So each edge corresponds to a pair of genes. Each node is a gene. And by colors, I'm indicating the gene ontology terms associated to each particular gene. So you might be asking whether the genes at both ends of an edge have something in common. For example, these two genes have the violet term in common. These two genes also have the violet term in common, and these two as well. And you might ask whether it's more often than expected. And for this, first, let's have a look at this particular graph of gene interactions and represent it in some other form. We'll represent it as a matrix. It's a binary matrix, contains zeros and ones. Each row is a gene ontology term. Each column is a gene. So the value of 0 is gray. The value of 1 is the colored solid box. And we have pairs of genes, which are indicated by the edges in this graph, which we represent by arcs between the columns of the matrix. So you may have a look that these two representations actually have the same information stored. This pair of genes has this dark blue term in common. This pair of genes has, for example, this violet term shared, and this violet term is also shared between these two columns, which are connected by an arc, and between these two columns, which are also connected. So you will notice that there are 3 edges in total, which have the violet term on both ends, so like in both of the interacting genes. You may ask whether it's more often than expected by random chance. How do we answer this kind of a question? Well, we need some kind of control or a background model. And for this, we are permuting the associations between the gene ontology terms and the genes. And we do it by taking the nodes in this graph and permuting them randomly. So there are 4 different permutations shown over here, where this set of annotations could go somewhere else in the graph, but we preserve the structure of edges, so things like cliques or hubs in the graph. So these representations could also be shown as these collections of arcs, and we could tally up together how often we see the same gene ontology term on both ends of an edge or of an arc, and quantify enrichment from these permutations and see how likely we are to observe this given number by random chance, by this sampling and permutation test. So this gives us at the end a list of enriched gene ontology terms, and this is what we observe for a set of gene pairs which were incidental to chromatin loops observed in high-C contact maps. These are the loops specific to GM12878 cell line. These gene ontology terms that are enriched are mostly related to chromatin organization, or they are the nucleosome, DNA packaging complex, protein DNA complex, nucleosome assembly, chromatin assembly, and so on. And what's interesting about these terms is that top three of them were also observed when we did a similar analysis, but for a set of genes, forgetting about their structure completely. So what we did is we did a similar analysis using an existing approach, using a new GEO tool, which is used for quantification of gene ontology term enrichment in a set of genes. And we found that only the top ones that we found using our methods were overrepresented using the other method, whereas the further ones were something which we found new as a new enrichment. And the converse was also the case. So the existing method, which doesn't take the gene pair structure into account, identifies lots of overrepresented gene ontology terms, which actually turn to have something in common to the type of these cells. These are the lymphoblastoid cell line GM12878, and the terms enriched using the existing approaches are mostly related to hematopoiesis processes, and they are kind of different sets of terms than the ones identified using our method GO. So to sum up, we propose a new method for gene ontology enrichment analysis, which takes into account the information on gene pairs that we look at. So this kind of information could come from the chromatin contacts or from some other spatial information that we have on the genome. So there are some more details in the PDF version of the paper. There is a link to the repository where you could download the source code, and feel free to reach me with messages or by email. Thank you.